Samantam, a faculty fellow at the National Brain Research Center, NBRC Manesar. On behalf of NBRC, I take great pleasure in welcoming all of you to this outreach event, which we are hosting as part of the India International Science Festival that will be held virtually from December 22nd to 25th, 2020. A faculty fellow at the National Brain Research Center. So with no further delay, I invite our director, Professor Neeraj Jain, to deliver his introductory remarks for the session. Professor Jain.
Thank you, Nivedita. It's my great pleasure to actually present two of our youngest faculty, Nivedita and Shubha, to give today's talk. And that too is part of IISF 2020, which is really a festival for the youngest people. We see a lot of students coming in, and so this is an event which is organized every year by Vibha in collaboration in one of the scientific departments, and you will see a film about IISF after my introductory remarks. So I am the director of NBRC, which is located in Manesar, a top institute for research on brain in health and disease. We are about 40 kilometers from Manesar on the from Delhi in Manesar on the Jaipur highway. And you can see a beautiful panoramic view of NBRC in the foothills of Shipalik mountain range. And I'll welcome you all to visit NBRC whenever you can and whenever this COVID-19 situation improves, to see for yourself what we do, meet with our faculty. The NBRC was established by Department of Biotechnology with a mandate to understand brain function in health and disease. To train manpower for advanced research and teaching in neuroscience, we are also deemed to be university, and we give Masters in Neuroscience degree as well as PhD degree. And we are also very keen on promoting neuroscience and science in general through networking among institutions across the country and among people. That's part of our mandate that's very dear to our hearts. You will, like I said, you will see a 3-minute short film about NBRC and learn a little bit more. And after that, you can learn more by visiting our website or sending emails, looking at our annual reports available on the website. And now I'll give you some thoughts about IISF. You are also will be looking at a film about IISF in a short while, a 3-minute film. So these are scenes from our NBRC stall from IISF 19. I attended 2 IISF festivals, and I must admit I was overwhelmed by the enthusiasm of people in last year in Calcutta, before that in Lucknow. It was just amazing how many curious minds come, visit our stall and other stalls, and learn about the process of science. They're so enthusiastic, so eager to know as much as we can tell them, that I realize this is an event which we should never miss and participate to a full extent. And I always urged my faculty, staff, students, postdocs, everybody to participate in IISF to as much as they can. So you can see here our live EEG demonstration last year because it was physical. This time it's a virtual event, like Nivedita said, and the advantage would be a whole lot of people from all over the country can participate in IISF in this virtual event. Of course, the disadvantage is that you can't experience it firsthand, an event like a EEG. Or this is another very moving picture for me from last IISF, where children from School for the Blind came and they wanted to feel how does the brain look like. So they're touching a model of the brain and trying to understand, and while one of our faculty, Dr. Laura Sen, is trying to explain about the brain to them. So this is what IISF is for me, a mega expo event, which is absolutely a huge success whenever I have been there. We always have a pre-IISF event. Last year we had an open day instead of that, because you can't have an open day because of COVID-19 pandemic. We are organizing these lectures, which will be delivered in a language by Nivedita and Puba, which everybody can understand. And you can see how many children have visited NBRC last year. They learned about our science through posters, through short lecture, and so on and so forth. So very exciting event for me. I will stop now so that we can now go on to watch NBRC film and then ISF film, and then it will be over to Nivedita for her talk. Thank you.
Thank you, sir, for your kind words. So, yeah, here's the film.
Popularly known as NBRC, was established by the Department of Biotechnology, Government of India, to understand the brain in health and disease. It is located in Manesar, just 40 kilometers from Delhi, and is a deemed to be university and an institute of excellence. The center has teaching programs leading to master's and PhD degrees. This premier institute has state-of-the-art infrastructure to conduct advanced research in different areas of neuroscience: cellular and molecular neuroscience, systems neuroscience, cognitive neuroscience, computational neuroscience and brain imaging, and translational neuroscience. To become a global center of excellence in brain research by 2025, NBRC has made significant breakthroughs in understanding disease mechanisms and developing world-class cellular and animal models for the neuroscience community. Core areas of research of the Institute are basic mechanisms of learning, memory and cognition, sensory and motor systems, neurodevelopment, effects of maternal malnutrition on the brain, neuroinfections, neurodegeneration, brain and spinal cord injuries and repair, brain tumors, and metabolic mapping of the brain. NBRC emphasizes studies of human brain with dedicated high-end EEG, MRI, and MEG systems. For basic neuroscience, mental health, and dementia, theoretical researchers develop models and technologies using data science and connectomics tools. The center is coordinating a national dementia science program involving 7 centers and hospitals from the length and breadth of India to understand incidence and prevalence of dementia in the country and the lifestyle and genetical factors that lead to development of dementia in old age. Another major area of research at the center is to understand and develop therapies for virus-induced encephalitis and other viral diseases, including Japanese encephalitis virus, Chandikura virus, HIV, Zika virus, and prions. NBRC has successfully established a stem cell platform in collaboration with Gurgaon General Hospital. This model systems developed at this premier biotechnology institute is widely used by researchers throughout the country and has helped to place India at par with international laboratories. A brain organoid model is currently under development. The institute also carries research in areas with broad social implications for children like dyslexia and autism. There is a center of epilepsy to develop a cure for drug-resistant epilepsy by bridging the gap between clinical and basic research, which is run in close coordination with AIIMS. NBRC has collaborative arrangements with many national and international institutes, including IIT Delhi, National Institute of Mental Health, NIMH, United States, National de la Santé et de la Recherche Médicale in CERN, France, and Forschungszentrum Joliet, Germany. This premier institute is putting all the energy in understanding the most complex organ of the human body, brain. इस वर्ष 22 से 25 दिसंबर 2020 के बीच भारत अंतर्राष्ट्रीय विज्ञान महोत्सव अपने छठे संस्करण के रूप में डिजिटल प्लेटफॉर्म पर अवतरित हो रहा है। देश में वैज्ञानिक सोच को बढ़ावा देने में तो इसका बहुमूल्य योगदान है ही, साथ में विश्व कीर्तिमान स्थापित करने। वैज्ञानिकों को ये विश्वास है कि वे हमेशा की भांति नए कीर्तिमान स्थापित करेंगे और देश का नाम रोशन करेंगे। इस वर्ष हम 5 नए गिनीज वर्ल्ड रिकॉर्ड बनाने का प्रयास करने के लिए तैयार हैं। एक वर्चुअल विज्ञान सम्मेलन में सबसे अधिक प्रतिभागी 22 से 25 दिसंबर का सप्ताह, समय प्रातः 11 से सायं 5 बजे। 2 एक साथ ऑनलाइन सन डायल किट असेंबलिंग, संपूर्ण प्रोसेस के लिए निर्धारित समय 22 दिसंबर को प्रातः 10 बजे से दोपहर 1 बजे तक। 3 सर्वाधिक व्यक्ति ऑनलाइन एक साथ हाथ धोएंगे, दिनांक 23 दिसंबर प्रातः 11 बजे से दोपहर 2 बजे तक। 4 एक साथ ऑनलाइन अधिकतर व्यक्तियों द्वारा रक्षक मास्क का प्रयोग, दिनांक 24 दिसंबर प्रातः 11 से दोपहर 2 बजे तक। 5 सीधी प्रसारित पौष्टिकता के बारे में वार्ता पर सबसे अधिक दर्शक एक साथ जुड़ेंगे, दिनांक 25 दिसंबर प्रातः 11 बजे से दोपहर 2 बजे तक। विज्ञान के क्षेत्र में जब कोई गिनीज वर्ल्ड रिकॉर्ड बनाता है तो वो अपने आप में महत्वपूर्ण है। आत्मनिर्भर भारत की कल्पना को साकार करने और विश्व कल्याण की दृष्टि से विज्ञान के प्रति जागरूकता उत्पन्न करने के लिए भारत अंतर्राष्ट्रीय विज्ञान महोत्सव एक अनूठा मंच है। अधिक जानकारी के लिए हमारी वेबसाइट पर जाएं www.scienceindiafest.org।
So that much for the introduction to the session. And now we proceed to the outreach lectures. The first one will be given by me, and my colleague, Dr. Dhruba, will follow. So, so the title of my talk today is Rhythms of the Brain. So, and I will want to start my talk by showing you this very familiar diagram that many of you, or most of you, would have seen in your high school biology textbooks. So this diagram shows the brain divided into discrete regions which perform discrete functions. Say, for example, the anterior or the front part of the brain that performs, that is necessary for concentration and planning, the back part or the posterior part that is responsible for vision, the lateral part that is responsible for hearing, and the lower back part that is shown in blue, which is a cerebellum that is responsible for coordination of movements. But the question comes is this actually how the brain functions? And the answer would be unfortunately no, because although this is a very good illustration that is very much simplified, this is not the actual reality. In reality, your brain looks something like this. So with innumerable number of nodes which are connected by innumerable number of connections. So these extensive connections of different regions of the brain is what enables us to perform the most complex of behaviors and experience the most complex thought processes. So for the generation of even the simplest thought or action, it is necessary that the brain actually coordinates or the interaction between different brain regions to carry out that thought or action. And for this high level of coordination, it is necessary that the different regions of the brain interact with each other and pass on information. And how does this information exchange happen between the different regions of the brain? That is a question that scientists have been trying to answer for several years now. And my talk will also focus on that. At this point of time, I want to introduce you to this really nice book written by Professor Georgi Bushaki, who is a neuroscience professor. And the title of the book is Rhythms of the Brain. So now you know where I got the title of my talk from. So this book presents a very nicely the current understanding of brain rhythms and how they enable functioning of the brain. And it is written in a very simple language that can be read by even non-scientists, and a good book for neuroscience enthusiasts who want to go back and read more about brain rhythms. So what is a rhythm? A rhythm is something that repeats itself periodically, just like the pendulum of the clock. And if you can recall your physics lessons in high school, so this motion of the pendulum of the clock can be represented as a sine wave, as it is shown here. And you can see this kind of periodic motion or oscillatory motion in many systems. And the brain is not an exception. So you can also see oscillations in the brain. So these periodic firing of neurons within the brain. Neurons are nerve cells within the brain that fire periodically, that get excited in a specific at a specific rate. And this periodic firing of neurons generates electrical activity, which can be represented as waveforms. And these waveforms are called brain waves or brain oscillations or neural oscillations, as I would be calling them. So Professor Bushaki actually puts it really nicely. He says, just like the clocks tick, the bridges and skyscrapers vibrate, neuronal networks oscillate. And now let me start with how these brain waves or neural oscillations were first recorded. So Hans Berger is a German was a German psychiatrist who was actually working on brain waves for several years. And in 1929, finally, he was able to record these brain waves by placing metal electrodes on the scalp of his patients. So these metal electrodes could collect some signals and display them something like this. And they were not sure where these signals were arising from, whether they were coming from the skin glands or from the muscles or from the brain. So to show that, what they did was he asked his patient to do a mental arithmetic task, and then the recording that they saw was completely unbelievable in that time. So the graph that you see, the bottom recording, shows a reference time scale of about 10 hertz. 10 hertz means there are 10 cycles of waves per second. And the upper recording is what was recorded from the subject. So the arrow that points upwards represents the time until when the subject was actually performing the mental arithmetic task. So as you can see, when the subject was performing the mental arithmetic task, the frequency of oscillations was much higher. You saw faster waves, which are smaller in amplitude. And the moment that the subject stopped doing the mental arithmetic task, the waves began to become slower, and the amplitude was larger. And this was approximately about 10 hertz, matching the rhythm of the time scale, reference time scale below. So this confirms that these waves were arising from the brain and not from any other sources in between the electrodes and the brain. So, Hansberger called it the alpha rhythm, that is the 10 hertz waveform that was clearly seen when the subject was mentally relaxed. He called it the alpha rhythm. And now we know that there is not just the alpha rhythm, but there are other oscillatory patterns in the brain named after the Greek alphabets in the order that they were discovered. So we have the alpha, beta, gamma, delta, and theta waves. And each of these oscillatory patterns have different frequency bands, associated with them. Like the alpha band would be 8 to 13 hertz, the beta band would be 13 to 32 hertz, and gamma would be 32 to 100 hertz. So, and these individual frequency bands seem to be associated with certain brain functions. Like we already saw in Hansberger's recording, gamma oscillations mediate or are associated with higher mental functions like problem solving, arithmetic calculation, learning, and perception. Whereas the alpha waveforms are seen when the subject is mentally and physically relaxed. So the slower oscillations, like the theta and the delta, seem to be, you can record them when the subject is in sleep or in altered states of consciousness. So what are these oscillations and where do they come from? So neural oscillations are generated by synchronized rhythmic patterns of electrical activity that are produced by neurons in the brain. So as you can see in this small graphic, there is a region on the brain which is red in color, and another region that is blue in color. So the neurons which are within the red region, when they fire, the synchronized electrical activity of all these neurons will be recorded from the surface or from inside, from near the cells, as the red waveform there that is represented there. And the neurons that fire collectively in the blue area, they give rise to the blue waveform. So this is how brain oscillations are formed. And the rhythm, the periodicity of these oscillations are basically they reflect the balanced interaction between excitatory and inhibitory neurons. So it's basically the interaction between different sets of neurons that generates this periodicity in the brain activity that we can record by different methods. Brain oscillations are present everywhere in the brain, and each brain region has a preferred oscillatory pattern. So we can, and then it's not just a single pattern, but some regions, they have several oscillatory patterns coexisting within them, and these different oscillatory patterns can interact with them. Say, for example, there could be a region which is purely having neurons that fire in the alpha frequency range. But for example, you have the parietal cortex that will have neurons that fire in the gamma frequency range as well as in the alpha frequency range. And the functions that they drive could be completely different, or they could drive functions where both of them have to interact and drive behavior. So also, the same as oscillatory pattern may drive different behaviors in different brain regions. So we can record alpha oscillations from the front, from the back, or from the side of the brain, but the functions that they perform in the front of the brain would be different from the functions that they would drive when they are present in the back part of the brain. And in general, faster oscillations seem to drive local networks, whereas slow oscillations seem to be mediating long-range connections within the brain. So how do we record these neural oscillations? We can record them at 3 different levels: the microscopic level, the mesoscopic level, and the macroscopic level. So at the microscopic level, what we can record is we can put needles into the single neuron, a single neuron or a nerve cell, and we can record the action potential of or the electrical activity of that individual neuron. And this is very similar to somebody having a one-on-one personal interview with a person. In the mesoscopic scale, what we record are called the local field potentials. So you're basically putting an electrode into the brain region, and this electrode is picking up the collective electrical activity of the neurons in that area. So, and this has a spatial scale of approximately a few thousands of neurons. So it picks up activity of a few thousand neurons, not just a single neuron. And this is, you can compare this to listening to somebody in a live press conference. And lastly, we have the macroscopic scale, which is similar to the EEG that we are recording today, which involves recording from a scale of a few 100,000 neurons, that is a specific region of the brain at a macroscopic scale. And if you compare this, then it is like listening to voices in an IPL stadium, for example. So then you can imagine the amount of noise that you actually get in the waveforms that you record with EEG. But the synchronized activity, so when you do an activity, the synchronized firing of the neurons actually make it possible to isolate the true signal from so much of noise that can be present in the background. And EEG is still being used in spite of such poor resolution because it is non-invasive. That is, you don't put in any needles or anything. It can be measured non-invasively, and it can be done without any pain in babies, in infants, in adults when they are in the alert state. Unfortunately, in humans, you cannot put in electrodes into single neurons or populations of neurons in alert individuals, in normal subjects, to understand brain function. So that is why EEG still stands to be extensively used in neurology and psychiatric research, as well as diagnosis. So why are we even studying neural oscillations? Why are they important? So neural oscillations constitute a key mechanism for controlling selective information transfer between brain regions. And if these oscillations are synchronized in time, then they even drive conscious perception, cognition, and higher mental functions, as I already mentioned. When you interfere with the physical properties of these oscillatory patterns, that is, you change the frequency, or if you change the phase of the oscillations, then you can impair behavior, that is, interfere with behavioral output, interfere with perception, or interfere with cognition. And if you enhance the synchronization of these oscillations, then you can eventually improve behavioral performance. This is also possible. And abnormalities in oscillatory patterns are associated with neuropsychiatric disorders. So it becomes more important to study them, to understand why these oscillations are abnormal in neuropsychiatric disorders and how we can correct these abnormalities. So, and therefore, neural oscillations could have immense therapeutic potential. So I will be this, for example, we can talk about deep brain stimulation that is applied for the treatment of movement disorders. So I will be addressing all these points in detail by showcasing 2 or 3 research studies that have been done very recently. So before I go into the research studies, I just want to give you some idea about some introduction about the terminology that I will be using to actually discuss these studies. The first one is phase synchronization. So consider these 2 regions, the red and the blue region of the brain, which are oscillating or basically firing at a certain frequency that is indicated by the red and the blue signals that you see here. So the red signal, the peak of the red signal coincides exactly with the peak of the blue signal. And the trough, that is the lowest point, the minimum point of the red signal, also coincides with the trough of the blue signal. So if you encounter signals like these, it means that these two signals are perfectly in synchrony. That is, they are phase synchronized, and they are also called in phase because the peaks of both signals coincide, the troughs of both signals coincide. So in the other case, if you consider the peak of the red signal to be coinciding with the trough of the blue signal, and the trough of the red signal to be coinciding with the peak of the blue signal, then it means that they are still in synchrony, in perfect synchrony, but they are out of phase or anti-phase. That is, there is a phase difference. If you remember your 12th standard physics, then you can say that there is a phase delay of 180 degrees between the two signals. The second term that I want to introduce is entrainment. So entrainment is basically trying to induce or trying to interfere with the intrinsic brain oscillation using external forces. So you can apply external rhythmic forces, like it could be as simple as flickering light stimulus or visual stimulus at a certain frequency, or it could be delivering magnetic or electrical pulses at a certain frequency so that they would drive the underlying brain oscillations to their external frequency. So say the figure that you see here, sorry, I'm not able to use my mouse cursor here, but the figure that you can see here that shows entrainment, you can see that the blue wave gets entrained. That is, the phase of the blue wave is being driven by the phase of the external, the black square wave that you see. So when the peak of the black wave comes, the blue wave also reaches the peak. So you're basically driving the rhythm of the brain to the rhythm of your external stimulus. So this can be achieved by using a technique called transcranial alternating current stimulation. So it might sound complicated, but cranium is your skull. Alternating current is the current that flows in your power lines, and transcranial alternating current stimulation means you're basically stimulating your brain tissue with the alternating current through the skull. And this is the device that is used for delivering TACS. And what you do is you basically put these electrodes on the regions of the brain that you want to stimulate and deliver alternating current at a certain intensity or at a certain frequency. So if the frequency of the stimulation matches the frequency of the underlying brain region, they would entrain the underlying brain tissue to their phase and frequency. Say you're delivering a frequency of 40 hertz, neurons that are underlying, which are oscillating already at 41 or 39 hertz, would align to a frequency of 40 hertz, and they would become in phase with the the stimulator's signal. So this is, and this will cause, so this entrainment is known to cause behavioral changes. So this is exactly what the study that I will present now will show. So in the study, this, the visual perception is what was investigated by the scientists. So this is for a group including Dr. Helfrich, and this study was published in 2014. So what they presented was a visual motion perception paradigm. So as you can see in the figure, it's called the stroboscopic alternative motion paradigm. So there are two dots that appear on the screen on diagonally opposite corners of the screen. And when they are presented in alternating appearance, then the person perceives it to be such that the dots are moving either horizontally or vertically. But basically, they are just appearing and disappearing. They're just blinking in their in the same position. So this perception of motion that is seen by the subjects can be actually be a good. It's a higher mental function that one can actually test. So the dot that is on the right side of the screen will be processed by the left side of your brain, and the dot that is on the left side of the screen will be processed by the right side of your brain. So if you have to see the dots moving in the horizontal direction, if you want this horizontal perception, then the images that are formed in the left and right cortices of your brain should basically be integrated into a single image. So you need oscillations from the left and right part of the brain to be talking to each other. They should be in synchrony to have this horizontal motion percept. Whereas in the vertical motion percept, you don't need the two regions of the brain to be integrating any information. So whenever the synchrony between the left and right hemispheres is higher, the subject perceives horizontal motion. And when the interhemispheric synchrony is lower, the subject perceives vertical motion. And this is a normal phenomenon. And so this is an example of how it works. So the dots are appearing in alternate positions, and you can perceive it to be either moving vertically or horizontally. This is the paradigm. And the subjects were supposed to report whether they saw vertical motion or whether they saw horizontal motion. So that's basically the task. And EEG was recorded when the subjects did this particular task. And as you can see, the red arrows that show on the skull, these are the two electrodes that were the electrodes of interest. And they measured the coherence or the synchrony between these two electrodes. And for different frequency bands, you have the delta, alpha, beta, and gamma oscillatory bands. And what they saw was for the delta, alpha, and beta frequency bands, there was no difference in the coherence between the two hemispheres for the horizontal and the vertical motion percept. Whereas for the gamma frequency range, they saw that the coherence was much higher for the horizontal percept than for the vertical percept, as you can see by the pink and the purple lines on the graph. So this again shows that you need higher synchrony, interhemispheric coherence between the two parieto-occipital regions for horizontal perception than for vertical perception. And the authors further, they wanted to see whether this was just a crude association that was there with the perception, or does the oscillation, that is this oscillation, the synchrony between the two oscillations, does it actually cause this perception? So the causal role of the oscillations for this behavior of perception, they wanted to test. And this they tested by using transcranial alternating current stimulation. So because the gamma frequency range was the most interesting one, they used 40 hertz TACS, and they put electrodes on the regions that they saw was showing interhemispheric coherence on the left and right side. And the subjects were invited for the experiment on 2 days. On one day, they received in-phase stimulation, like I already pointed out. And the second day, they were given anti-phase stimulation. That is, for in-phase stimulation, they received exactly the same signal on the both left and right hemispheres. Or for anti-phase stimulation, they received two signals which are 180 degrees out of phase between each other on both sides. So the hypothesis is that if you give in-phase stimulation, then there should be higher coherence between the two hemispheres, and this should lead to more horizontal perception by the subjects than vertical perception. And with anti-phase stimulation, the subjects should be seeing more vertical perception than horizontal perception. So this was the hypothesis. And as you can see, there were different blocks of recordings that were made with the EEG. So RS means resting state. So resting state blocks of 3 minutes each were recorded when the subjects were not doing the task. It was just resting state EEG that was measured. They was just staring at a dot that appeared in the center of the screen. So, and sham and post stimulation sessions were 10-minute duration each when there was no real TACS that was given. During the stimulation, the 20-minute stimulation session, they received 40 hertz TACS, 1 milliampere intensity for 20 minutes. And one day they received in phase, the other day they received anti-phase stimulation. And here are the results. So in this graph, the top graph, what you see is the line, the dotted line that you see in the middle, around 49%, indicates the normal ratio, that is without any stimulation. That was the baseline for the number of percentage of times horizontal movement was perceived versus the vertical motion. So any dot that is above this dotted line, any point that is above the dotted line indicates that there was more horizontal perception, and any point below the dotted line indicates that there was more vertical perception. So as expected in the sham and the post-stimulation conditions, when there was no TACS that was given, you can see that it was almost about the same. That is, there is no change in the horizontal or vertical perception. But with 40 Hertz TACS, with the in-phase stimulation, the subjects showed an increase in the horizontal perception than vertical perception. And with anti-phase stimulation, they observed more vertical perception than horizontal perception. And this was on the behavior side. Now they looked at the EEG to see what was actually causing this change, difference in the percept. And they looked at the coherence again between the two hemispheres. And they again, as expected, they saw that the coherence between the two hemispheres was increased with in-phase stimulation than with anti-phase stimulation, as you can see. So the green line is in-phase stimulation and the orange line is anti-phase stimulation. So this shows that there is a causal relationship. You can change the oscillatory synchrony and also change, and with that you can change the performance of the subject or the perception of the subject. So this is with respect to normal oscillations. And I just want to give you an interim summary at this point. I just want to say that neural oscillations are generated by rhythmic activation of excitatory and inhibitory neuronal assemblies. They are crucial for information transfer within the brain. As we saw, the information transfer was occurring between two hemispheres of the visual processing areas. They play a causative role in perception and cognition and probably some other mental functions as well, which we have still not explored. And non-invasive brain stimulation methods such as TACS can modulate neuronal oscillations, and thereby they can also alter behavior. So that is about normal or physiological oscillations. Now we go into abnormal neural oscillations. As I already mentioned, almost all neuropsychiatric disorders have been known to have abnormalities in the neural oscillatory pattern. For example, epilepsy patients have shown faster ripples in hypersynchrony, this enhanced synchronization of firing of neuronal assemblies. Parkinson's disease patients show an enhanced beta oscillatory pattern. And schizophrenia and depression patients show a much lower gamma oscillatory pattern. So like mentioned earlier, these rhythms or oscillatory patterns have been associated. They only have a correlational association in these diseases. And we still have not studied whether these oscillations, abnormal oscillations, are actually causing certain symptoms of the disease, or are they just an epiphenomenon, so that whether they are just occurring as a side effect of the symptoms that are happening, the changes that are happening in the brain. So increase or decrease in amplitude of oscillations is what we have studied extensively till now, but just an increase and decrease of amplitudes is just too simple to explain the complex behavioral changes that happen in manifesting symptoms of diseases. There are more complex relationships that seem to exist between oscillatory patterns, such as cross-frequency coupling, but I will not go into further details. It's more complex than we think it is. So my favorite topic is movement, is in neuroscience, is movement disorders, and that is what I study. And Parkinson's disease is one of the most common movement disorders that many of you would have heard. You probably have a family member or a friend who had Parkinson's disease, or you would have just heard about it somewhere in your textbooks. So Parkinson's disease is a neurodegenerative disorder, which means there are loss of neurons that secrete a specific chemical called dopamine in the basal ganglia of the brain. So in this image of the brain shows where the part, that is the basal ganglia part, is really present. And basal ganglia, the function of this basal ganglia is movement control. And it basically acts as the brake in your car. So you can imagine what happens if the brake in your car fails. So and that's exactly what happens when the basal ganglia fails. It has a very important function. So patients with problems with the basal ganglia end up having really disturbing and disabling movements is because the dopamine releasing neurons are specifically affected. You see less dopamine, like the scan image that is seen lower corner. You can see the dopamine levels of the dopamine levels in the basal ganglia of normal subjects is much higher than the dopamine levels of Parkinson's disease patients. That's on the right side. So you can see that there's a really low amount of dopamine that is present in the basal ganglia, especially, and this gives rise to symptoms such as slowness of movement, rigidity, tremors at rest, and postural imbalance, among several other non-motor symptoms as well. And logically, what you would do is just give them dopamine supplementation, and that is what we still do, and that is the gold standard treatment until now. But it does have a lot of side effects, because this is not mimicking the dopamine that is naturally being secreted in the brain. So this is something that you give externally, and the brain reacts differently to this extrinsic dopamine as compared to the intrinsic dopamine. And another treatment modality is the surgical option, which is the deep brain stimulation that has been revolutionary in the treatment of Parkinson's disease over the past decade or so. So in deep brain stimulation, what happens is you're putting electrodes into the basal ganglia of the patient and connecting it to a pacemaker-like device. It's a pulse generator that generates signals of very high frequency, that is about 130 to 160 hertz of 130 to 160 hertz signal is what is delivered to the basal ganglia regions. And by some mechanism that is still unknown to us, it happens to dramatically fix most of the, many of the symptoms of Parkinson's disease. Doesn't fix all the symptoms. It does have side effects, but the symptom that is most problematic for the patient seems to respond best to deep brain stimulation. So this relates to oscillations. So Parkinson's disease patients also have abnormal oscillations, and this has been studied in the last 10 years extensively. So this was a study in 2008 published by Peter Brown's group, which shows that is in this graph, the left side of 0 is the duration when there was high frequency stimulation kept on. And the right side from 0 to 200 seconds is the duration when the stimulation was switched off. So as you can see, and this is the frequency spectrum, that is the oscillatory activity that was recorded using local field potentials, that is electrode present in the brain, and you're recording from the basal ganglia. And what you can see, the stark difference is the red patch that occurs about approximately 25 hertz of the frequency band, and this belongs to the beta band. Right, so the beta frequency seems to be occurring, that is at higher intensity when the stimulation is switched off, and this beta band seems to be, and when switched on, this increased power is gone. So basically, people thought that this beta band could be the bad oscillation in Parkinson's disease, but they wanted to see how it actually correlates to motor symptoms of the disease. So they did the same thing. What they did was they gave high frequency stimulation, and then they recorded local field potentials when the subjects were at rest, as well as when they were making a small movement. And they wanted to see how the movement pattern was affected when the stimulation was on versus off. And they saw that when the stimulation was off, as and when the beta oscillatory pattern increased in amplitude, they also saw that the movement amplitude decreased. That is the slowness of movement. The motor symptoms came back. They were worse when the beta oscillatory pattern actually emerged. So this showed a clear correlation of the beta oscillatory pattern to the motor symptoms of the patient. And now the same group has started working on something called, so this concludes that beta oscillatory power in the basal ganglia is higher in patients with Parkinson's disease, and this is probably the bad oscillation that we want to control in these patients. And secondly, with deep brain stimulation at approximately 130 hertz, the beta power decreases and is associated with improvement in motor symptoms. So, like I said, this group has worked on something called adaptive or closed loop deep brain stimulation. So if we know that the beta oscillation is the bad oscillation, why do we have to really continuously keep stimulating the basal ganglia? So in the conventional DBS, the patient receives stimulation continuously 24/7 until the doctor stops it. But this is not what we really want to have. We want the stimulator to turn on when the beta oscillatory power is too high. And we want the stimulator to turn off when the beta oscillatory power is not so much. So this is what they developed. They developed an algorithm, a system that will actually record the LFP signals simultaneously as the stimulation is being given. And this LFP signal, that is a local field potential signal, will be then filtered to the beta frequency band, and the power of the beta band will be computed real time. And you can actually see that they fix a threshold. So when the beta power goes above this threshold, then the stimulator will turn on, and which will decrease the beta power, bring it back to normal. So the figure on the right actually shows you clearly, the top one shows the beta burst, the amplitude of the beta oscillation, and the lower panel shows the DBS stimulation pattern. You can see that the stimulation starts exactly when the beta power is at the maximum, that is, it has reached the threshold. And then when the beta power decreases, then the stimulation stops. And then this goes on. You can see the arrows that show when the beta burst is actually peaking or reaching the threshold. So this kind of adaptive stimulation pattern seems to be equally effective as the conventional DBS. And the efficacy of adaptive DBS seems to be achieved by just half the stimulation dose of conventional DBS, which means that with the lower dose, you have lower side effects and you have a longer battery life of the stimulator. One of the major concerns of the DBS is that the battery of the pulse generator that is implanted under the skin of the patients on the chest, the battery has to be changed once every 2 or 3 years. And with this adaptive DBS, you can the battery life is probably going to last for about 5 years or so, which means the surgery is postponed. You have lesser surgeries, and the cost is also less. So this seems to be a much better working pattern and a great level of improvement to the conventional DBS that patients have been receiving till now. So, but this is still not approved for clinical use. It is under clinical trials. As you can see, the first paper was published only 3 years ago in Brain, and people are still working on larger scale trials, clinical trials. So, we saw that oscillatory patterns are abnormal, and you can actually use certain oscillatory patterns to identify the abnormal rhythm, and also see if it would fix the certain symptoms of the disease. So information transfer oscillations, and these neural oscillations seem to be causally linked behavior. This is the first study that I showed you, where physiological oscillations can be modulated and to alter perception. Neuropsychiatric disorders are associated with abnormal brain oscillatory patterns, such as in Parkinson's disease, which is a good example that I have shown you. Then identification of abnormal oscillatory patterns with diagnostic value is essential. A very good example was the beta oscillatory pattern that I just discussed. So identifying this abnormal oscillatory pattern has a diagnostic value. And last but not the least, we have non-invasive modulation of brain oscillatory patterns, which show great promise in the treatment of movement disorders. So if we can replace the function of what deep brain stimulation does with non-invasive brain stimulation techniques, then that would be really awesome, because it is just less than 1% of Parkinson's disease patients who are eligible for undergoing DBS surgery. And if we can actually make achieve the same goal of controlling the brain oscillations or modulating the brain oscillations by using non-invasive techniques, then that would be a breakthrough. So that's about brain oscillations and an overview of how, or overview of what exactly brain oscillations do and how they are modulated and how they are different in altered in disease states. Now, I just want to tell you what we at NBRC do. So this is the Human Motor Neurophysiology and Neuromodulation lab that I run at NBRC. It's a very new lab that we have established, very few months old now. And these are the few research questions that we are trying to address. The first one, how does brain control movement? Sounds very simple, but the more you read about it, the more you study it, the more questions come up. And there are so many unsolved mysteries in how the brain controls movement in normal subjects, in just you and me. And what mechanisms in the brain go wrong in patients with movement disorders? And thirdly, how are the oscillatory patterns of the brain altered in different movement disorders? And finally, can we improve certain disease symptoms by restoring the normal oscillatory patterns using non-invasive brain stimulation techniques? So to answer all these questions, we cannot just use a single technique. We have to use a multimodal approach, which we do. We have the transcranial magnetic stimulation, the magnetic resonance imaging, MRI, and MRI-guided neuronavigation to localize a stimulation to a specific region of the brain. Then we have EEG that is compatible with the TMS system, so we can give magnetic stimulation and see what happens to the brain oscillations. We have EMG and also the transcranial electrical stimulation. So by integrating all these methods as and when appropriate to answer the research question, we are trying to find solutions or answers to our research question. And finally, this is my small team that we have just formed, very small and early and young. We're hoping to expand soon. And thank you all for your attention. I now hand over the mic to my colleague, Dr. Dhruba Singh. Dhruba?
Yes. Thank you, Nivedita. Thank you for giving me such a wonderful talk. Today, I'm going to present use of Drosophila model in the neuroscience. So Drosophila melanogaster is commonly known as a fruit fly. It is used in the various biological research because of the many of the characteristics, such as they're inexpensive to culture, they have a very short lifespan, and they produce large number of progenies, giving large number of samples, and they have only 4 chromosomes. So because of that, the genetic process are very simple. However, they also have a small and simple brain which activates high order functions such as memory and learning, allowing us to study the neuroscience in the fly. Because Drosophila, various life cycle of Drosophila are used in various biological researches, so I'll briefly explain you the life cycle of Drosophila. Drosophila is homometabolous, which means they undergo complete metamorphosis. So after the egg is laid, it undergoes embryogenesis, and after 1 day, it becomes larvae. So the first larvae is called as the first instar larvae. Then after 1 day, the first instar larvae molds to form the second instar larvae. Then after 2 days, it forms the third instar larvae. It becomes quite bigger in size. Then during the third instar larvae, it secretes the cell around itself to form pupae. And after 4 or 5 days, it becomes dark. And then after that, it metamorphoses into adult. And the adult fly survives for about 80 to 90 days. So if you're planning the experiment, then you can get new samples every 5 or 6 days. So Drosophila are basically cultured in the bottles or the vials. So here you see that in the bottles there are food. So these food are basically called as a ferment media. That basically consists of the maize powder, agar, yeast, and the sugar. And there are basic tools that are required to observe the Drosophila. This is the dissecting microscope or a stereosome microscope. Then this is the fly pit where you can observe the flies. And here is the carbon dioxide needles, where you can inject the carbon dioxide into the vials or the bottles to anesthetize them. Then this is the morph, where you can put the dead flies. Then you see there are brushes, and this is the marker. So you need very basic tools to observe the flies. You don't need very sophisticated instruments. And because of this 100 years of the research on the Drosophila, various mutations lines are available nowadays. But the first of the mutation in Drosophila is called as white eye mutations. And as you see here, the eyes of Drosophila are normally red, but in the white eye mutation, the eye is white in color. And this white eye mutation is X-linked recessive mutation. And the work on this white eye mutations led to the discovering of the chromosome basis of inheritance, which led to the Nobel Prize to Thomas Morgan in 1933. In addition to these spontaneous mutations, the mutation in Drosophila can be induced by X-ray. So the student of Morgan, Hermann Muller, induced large-scale mutation in Drosophila using the X-ray. And because of his work on the X-ray, he got a Nobel Prize in 1946. And this is the curly wing in the fly. In the normal fly, you see that the wings are straight, but in the curly wing, the wings are curved upwards. So the mutation in the flies can be induced by the radiation. And moreover, the mutation can be also induced using the chemicals. Here is the mutation of the Drosophila induced by the nitrogen mustard. Here you see the pupae of the fly are shorter as compared to the control. So one of the large-scale mutagenesis was performed in 1979-80 in Heidelberg by Nusslein Bohart and Eric Michels, and this screen is basically called as a Heidelberg screen. And in this they use ethyl methane sulfonate to induce mutation, and they identified about 600 mutations affecting about 200 genes. And this is an example of some of the mutations. The first of this is the mutations of echoid. And here you see the structure of the head, of the larval head, is not properly formed in the echoid mutation. At the same time, in another mutation called Staufen or Stau, the interior and the posterior part of the brain is interior and posterior of the larvae is not. So Drosophila has also been instrumental in discovering various biological phenomena. And one of the mutation is the mutation of the Hox genes. The Hox gene is the cluster of the genes which are basically involved in specifying the body plan. And in the mutations of one of the Hox genes called UBX basically transforms the halteres, which is basically a balancing organ, into to form the wing, and the mutation also leads to formation of the two thoraxes. Work on the UBX genes led to the Nobel Prize in 1995 with Lewis, along with Nusslein Bohart and Eric Michels. In addition to this, there are multiple pathways and the mutation lines that has been found to be in the human also. One of these pathways is called wing or wing pathway, in which there's loss of the Drosophila wing, and in another mutation called as notch, there's a small notch in the wing of the fly. In addition to this, there are some multiple other mutations like Hatchcock mutation, cell death pathway, insulin selling pathway, which are basically conserved between the human and flies, indicating that the Drosophila model is relevant to the flies, relevant to the human space. One of the interesting study that has been discovered in the Drosophila is the discovery of the biological clock or a circadian rhythm. Most of the animals, they follow 24-hour day in the life cycle. And the molecular basis of this circuitin was first discovered in Drosophila. So there are two proteins, por and a thym, which basically cycles between the day and the night. So por and a thym, basically the transcription of these two genes starts during the daytime, and it builds up. Then in the evening, at night, it locates into the nucleus. Then it blocks the night. It binds to the clock and cycle, and the blocks its transcription. So the work on this clock and cycle, por and thym, led to the Nobel Prize to three of the scientists, Jeffrey Holt, Michael Rosbus, and Mike Yen in 2017. So because of so many years of the research in Drosophila, various tools and techniques has been already developed, such as the US GAL4 system, electroretinogram, neuromuscular junction, electrophysiology, and thousands of the nuclear lines has been generated, and thousands of the RNAi logs has been spared. These logs have been created, which we can basically any of the scientists can just order from the stock center and use. Moreover, there are also various protein trap lines which can be used to study the localization of protein. So I'll briefly explain what is the US GAL4 system. The US GAL4 system allows expression of gene in a tissue-specific manner. Here, in the US GAL4 system, you have one line of fly which expresses GAL4 in the tissue-specific manner. The tissue can be in the eyes, in the brain, or in the abdomen, or in the leg. And you have another line which is called US, where gene has been grown after the US. So when you cross these two flies, the flies in the first generation have both GAL4 and the US line. So the gene of interest will be expressed only in the tissue where the GAL4 is expressed. So in this way, you can either express the gene of interest, and if you are expressing the inverted repeat, you can downregulate the genes, or if you're expressing the JP, you can basically observe the JP in that specific tissue. So using this US GAL4 system, various neurological disorders has been generated in the flies. So one of the diseases is the Huntington disease. As you see here, the Huntington disease is caused by the expansion of the polyQ repeats in the Huntington gene, and it causes movement disorder and dementia and the loss of the neurons. So the humanized form of Drosophila expressing the polyQ repeat with 108 repeat shows the loss of the retinal tissues here. So various other lines has been already generated in the flies, such as Alzheimer model expressing Abeta42 is already available, and Parkinson model expressing alpha-synuclein is available, and LS model expressing the TDP43 are already available. So in our study, in one of our study, we used Drosophila to overexpress ataxin-3, expressing 78 polyQ repeats. And expression of ataxin-3 with the polyQ repeat causes a degeneration of the eyes are rough, and there are patches over eye surface, which is caused by the large-scale cell death. And we found that overexpression of protein called DMYC suppresses the rough eye phenotype. The eyes are red in color, they are more similar size as wild type. And we found that the suppression of this eye phenotype is due to the preventing the sequestration of the CDP protein, which is a transcription factor, and which is transcription factor, which is downregulated or basically sequestered in certain acute condition, disease condition. Moreover, we also found that the isolated form of histone H3 are increased in the flies with the DMYC as compared to the 70IQ. So our study indicated that the modulation of DMYC pathway can be used to suppress the polyQ disease. In addition to this, Drosophila can be used to model various neurodevelopmental disorders. There are various neurodevelopmental disorders that's been already been modeled, like the Fragile syndrome with the knockdown of FMR1 gene in the flies. And there are also other genes which are involved in autism, like the neuroxin and the neuroglin. So Drosophila neuromuscular junction is used to study the synaptic morphology of and you see that these are basically the synaptic buttons of the motor neurons in the larval body wall muscles. So they are basically branched, and then this circular-like structure are basically the synaptic buttons. So in one of our studies, we found that knockdown of CDIPD, which is involved in the 16p1.2 deletion syndrome, which causes intellectual disability and autism, we found that the number of synaptic buttons are significantly increased, whereas in the PAM57, we found that the synaptic buttons are reduced in number. So indicating that the abnormalities in the synaptic buttons or active zones can contribute to the neurodevelopmental disorder. And the Drosophila can be also used to study the dendritic arterization, because in many of the intellectual disabilities patients, the number of dendritic ardors are quite reduced. So in Drosophila, we can use class 4 dendritic neurons, which is basically a peripheral neurons, to study the branching pattern of the dendrites. One of the important thing about use of Drosophila is that this kind of patterning can be observed at the cellular level in light imaging. So in this study, we knocked down the KCD13, which is again one of the genes involved in 16p1.2 deletion, and we saw that there is a decreasing number of the dendritic arbors. To check how we reduce, how to check the number of the dendritic arbors, we basically do a circle around the center of the soma of this cell and of this neuron, and then we draw a circle around 25 microns, and we counted the number of those dendrites which are touching at the center at this circle. And then we found that the KCD13, there's a reduction in the level of the dendritic arbors, but at the same time, we also found that TAUK2 leads to the increase in the number of the dendritic arbors. So this also indicates that abnormalities in the number of dendritic arbors may also contribute to the neurodevelopmental disorder. And Drosophila is one of the wonderful tissue to model the neurodevelopmental disorders, because the eyes are quite easily visible under a dissecting microscope. And you see here that in one of our studies, we used Drosophila eye to knock down some of the genes, like the CDP20 or a DLG1, which are involved in the 3q9 deletion syndrome, which also causes autism and intellectual disabilities. So knockdown induces roughening of eye, and we try to check what is the cause of the roughening of eye by observing the ommetidial cells by staining with the DLZ. Usually the fly eyes are very kind of soaring in structure that looks like a flower, but in the flies with a disease, you see that the structure is lost, many of the cells are dead, and the orientation of this ommetidia is different as compared to the control. And furthermore, we wanted to check what is the cause of this loss of neurons. Is it because of the cell death or the defect in the cell proliferation? And we found that this defect is due to the increase in the level of cell death as compared to the control. One of the hallmark of the intellectual disabilities or autism is that they have defective memory and learning. So Drosophila can be also used to study the memory and the learning. The memory and the learning in the flies is basically performed by one particular organ, this organ, which is called as a mushroom body, which consists of about 2,500 neurons. So to study the behavioral, to perform the behavioral studies, it needs one session of training and another session of testing. So flies are basically introduced into a tube which consists of a copper grid which can be electrifiable. And then after that, the flies are given the electric shock by introducing one odor which can be octanol. And then after some rest, the flies are given another odor without any electric shock. Then in the testing stage, the flies are introduced into a T mesh and allowed to choose between the two odors, either octanol or the MCH. So usually the wild type flies, they basically choose the odor which has not been associated with the electric shock. But in the flies with the mutations or the knockdown of genes involved in the autism or intellectual disabilities, the fly basically 50% of the flies, they remain in each of these tubes. And then the defect in the memory and learning is basically affect the habituation. Even in the fMRI patient, there's been found that there's a defect in habituation with the sound stimuli. So basically, habituation is a fundamental forms of learning where the organisms learn to respond to the meaningless stimuli to decline over time. So in the flies, we can use light of jump habituation to study this phenomenon. So here, the flies is illuminated with the infrared light. So in the normal flies, the fly understands that the infrared light, which is given continuously, are meaningless. So they started to reduce their, they started to have less jumping and the vibration of wing started to reduce. But if there's a mutation or the knockdown of genes involved in autism or intellectual disabilities, this vibration or the jumping of the flies keep happening for a longer period of time. So in one of the studies done by Pankova et al., so they performed the habituation studies with about 286 genes involved in intellectual disabilities, and they found that about 100 of the genes have a defect in the habituation. And another aspect of the autism is that they have a difficulty in communicating, and then basically do not respond to the social cues. And Drosophila can also be used to study the social interactions. To perform the social interaction in Drosophila, basically the Drosophila are put inside a triangular glass slab, a particular triangular glass slab, and the flies are basically introduced from here. And then they are tapped once. Then after that, it is waited for about 30 minutes to see whether how the flies behave. In the wild type flies, most of these flies go up into the tip of this or the vertices of this glass slab, and most of them congregate in close proximity to each other. But the flies which has a defect, which has a mutation or the knockdown of genes involved in the autism, these flies basically remain throughout the glass slab. Finally, Drosophila can be also used to study the repetitive behavior, as the repetitive behavior is one of the symptoms of autism. So repetitive behavior in the autistic patient can be simple as a hand clapping, can be dangerous as banging of the head on the wall or the body rocking. And Drosophila grooming basically is basically recapitulate this repetitive behavior. For example, see here, the Drosophila is basically grooming his head with his front leg. And in this video, you can see that Drosophila is basically cleaning his wing and hind leg. So this kind of activities, Drosophila spend about 30% of the time grooming their head or their body. But if there are some mutations or the knockdown of genes, then this can affect the grooming behavior, and they can undergo excessive grooming. So with this, I like to end my talk, and then I like to thank India International Science Festival, IISF, the organizers of IISF and my parent organization, National Brain Research Center, for giving opportunity to interact with you all. Thank you.
So yes. Dhruva, I'm just having a look at the YouTube channel, and I do not see any questions that have popped up yet related to your talk.
Yes.
But yes, nothing that came up during the talk as well. So I think, yeah, so if there are questions, maybe you can just try to answer them right away in the chat box, like I did.
Okay.
Yes.
Okay.
So yes, so this will mark the end of our outreach event this afternoon. And thank you all for your participation, and have a great evening. Stay safe and goodbye.
Thank you.